Hello, I am Mehrad Hajiaghabozorgi from University of Minnesota, and I'm going to present this my poster to you. So the title is Models for Simulating Genetic Interaction from Real Human Genotype. So it's basically my method is trying to simulate genetic interaction for GWAS data sets. So GWAS or genome-wide association studies has been used to find association between loci and traits from human genotype data. So one focus of GWAS researchers have been on finding genetic interaction from GWAS data. So there is an issue for that. So if you develop a method, a GWAS method for detecting genetic interactions, you really you feel the lack of a comprehensive standard and collection of genetic interaction with full coverage to cover every possible snippers. So you may use real data, but you're not sure that every false positive is actually false positive. So another approach is using simulated data sets with which we know every genetic interaction within that and use that for evaluating our method. So to date, most of these simulation methods have been focused on simulating a single interaction or a pair of a few genetic interactions which are isolated from each other. But what we are interested and more interesting for GWAS researchers is a structure like this graph with in which like a hub SNPs having interaction with multiple other SNPs. And this has been observed in model organisms that this happens. So our methods get some input from user, which are minor allele frequency, joint minor allele frequency of SNPs involved in interactions, odds ratio, which reflects a strength of interaction. And also we have like real genotype data. So we, based on those inputs and genotype data, we choose candidate SNPs that can satisfy minor allele frequency criteria. And then we compute scores. I'm going to more detail about that. And based on those scores, we have a random sampling, weighted random sampling process to draw samples to be in the case group. So the score is computed like this: alpha, which is base in effect size, and beta is interaction effect size, which is added to a sample for each interacting allele in its genotype. And we also have a Gaussian noise term with mean 0. So there are two constraints to be satisfied. One is like odds ratio formula and equation. Another equation is for keeping the number of samples in the case group equal to half of the population. So based on these two constraints, alpha and beta is computed. So we use bridge, a method developed in our lab to compute interaction scores and evaluate our method after simulation. So our simulation has two parts. The first part is checking if our method can successfully simulate single interactions. So we use 6 parameter sets, which is combination of 2 low and higher minor allele frequency range, and also 3 different odds ratio. So you can see, and we use sensitivity plot to show the results. Sensitivity plot is actually shows what proportion of simulated snippers have interaction score or greater interaction score to the point on x-axis. So as you can see in here, basically higher odds ratios and higher minor allele frequency ranges yields a better signal and higher interaction scores as expected. So, and also we have ROC plot showing true positive rates versus false positive rate, which actually measures like how the rate of false positives in our method. So you can see again higher odds ratios and higher false positive, higher minor alpha frequency ranges gives better distinction between simulated snippers and non-simulated snippers. So based on this, that we can say we have successfully simulated a single isolated interaction. So we can move on to multiple interaction with Hubble SNPs. So for this simulation, we fixed odds ratio and minor alpha frequency, and we focused on number of interaction with the hub. So we chose 1, 3, 5, 10. So our framework rejected 10 because it couldn't satisfy the conditions and these two constraints that I talked about. So, and then we have same plots for multiple interactions. So you can see still simulated interaction are detected and that they are successfully simulated. Although I should mention that the case with one interaction has lower interaction scores, because in cases with 3 and 5, marginal association with Hubble SNP, but between Hubble SNP and trait is gained, and it's reflected in all the snippers, simulated snippers. But still, we have almost perfect ROC curve. So there is a almost perfect distinction between simulated and non-simulated snippers within each simulation. So thank you for paying attention to my presentation, and bye.